Hello everyone, how are y'all doing today? Welcome on in. Uh-oh, there's the munchie. And happy 4th of July, y'all. We're going on a nature walk today. Yeah, we went all the way to Princeton. There's apparently a fancy bridge here that we're gonna be checking out. Is that true, Munch? We were looking up things to do in New Jersey. I came across this park in Princeton, New Jersey, and allegedly there is a famous bridge here that people come out to just for that part of the hike. So we're going to explore nature and we're going to find this bridge. Yeah, we drove out to Princeton today for this IRL excursion. I keep forgetting, y'all, to bring some vials on these trips for samples that we can look at underneath the microscope, because I'm looking at these lichens on this tree. I'm like, oh man, like we could probably find some tardigrades in there. So here's some on this tree. There was much more populated ones earlier on, and that one had like a ton more lichen. But if you scrape those off a tree, put it in some water, or even put them through like a thin mesh and run water gently through it, easy way to collect tardigrades. The easiest, Chad, is get a ton of those lichens, and then essentially you make a very gentle sieve, very small sized hole mesh, like a couple of microns in size, and then run the water gently through it, and then you'll end up getting a bunch of tardigrades. We got some raspberries, it looks like.
I don't believe those are raspberries.
Oh, what are they then?
Not sure. Possibly mulberries, but even that, I don't think that's right. I don't recognize it as a raspberry though.
They are Princeton berries. So, now if y'all can tell, there's a lot of openings and crevices and nooks on this tree. That probably was eaten at some point by termites, but I'm betting if we were to crack it open, we'd find some ants in here. So probably some carpenter ants inside of this. That's usually what happens, is the termites start chewing up the wood, and then the carpenter ants come on in and tear up the termites and move in. When we've cracked open, not here in Jersey, but down in Florida, like rotting wood with those holes in it, usually it'd be like 80% of the time you'd find ants in there. Someone is very excited, Chad. We are near the Princeton campus. I've been here once before, not on this trail. We actually did a couple of interviews at Princeton. One was with a fruit fly neuroscience area, and then another was with the biology department, and that one was with mosquito research. And fun fact, the mosquito lab here, you got to feed the mosquitoes with your body. That was the regulation at the time, where when you were being hired, it'd be like, hey, I'm not saying you can't be hired if you don't agree to feed mosquitoes with your body, but you might not be hired if you don't agree to feed mosquitoes with your body. Because you were infecting with different people, so like, I might feed the mosquitoes and the next day someone else might feed the mosquitoes and we might have two different blood types. You can imagine a situation where you're going to have different blood types for a mosquito, that, because it's eating two blood meals. And my question was, is that going to give a different result versus if you just feed them with one blood type? And she was like, yeah, we don't really think about that. So, two other ways to feed mosquitoes in a lab. One is synthetic, so you give them cow blood, and what you do is you make tubes in their feeding area with a thin film. You then put a heat source next to the blood, as well as a CO2 emitter. And so that's like a very artificial system, but they'll feed on it. And you can get the same kind of cow blood in. The other thing you can do is put a mouse to sleep, shave his belly, and put the sleeping mouse, into the mosquito bin. The mosquitoes feed on the mouse. Once you're done feeding, you take the mouse out. The mouse wakes up, is totally fine, and then they feed. And they're totally okay. Mice do not have an allergic reaction to mosquitoes. They don't have a reaction to the blood meals, because the reason we get itchy is because a mosquito spits into you. That's the your itchiness that you get. And so the mouse immune system doesn't recognize that as foreign. That's, I think, probably the cleanest way, Dacum, because then you can get the exact same genetic line of mouse, and then the blood genetics is the same thread every time. When I was interviewing at that lab that had to do the person blood meal one, they told us, they're like, if you do it enough, you get used to it. Like you stop reacting, you build up a tolerance for it. So Golganak, that is apparently a possibility. I don't know if that's for everyone. If that's a person-specific thing as well, because, right, immune systems vary. But as maybe. Oh, I bet we've got some carpenter ants in this, y'all. Look at how much this tree is just, like, destroyed. Oh, and yeah, it looks like there's some ant activity outside of it, too. That's the bridge. See, there's another bridge, too. Yeah, so this is the bridge, guys, that we came for. Here we go, chat. Here we go. But also a beautiful view. This is the part where the streamer falls in the water. It's a nice shot, too. Why did y'all drive an hour? Oh, we drove an hour for this 10-foot bridge. Well, she's thinking about checking out this bridge, y'all. Hi, Munch. Whee! Look at that smile, y'all. Look at the devious little smile. Yes, in the grossness. Dacum. In the gross. See? Then we go boing, boing, boing. Oh, the gimbal's keeping us steady. Wow! There are a couple of trees that we've seen fallen over that are kind of born into. If we were to crack some of them open, I think we'd see some kind of carpenter ant colony. Easier to do, though, with like a actual set of like screwdrivers or hammer or an axe or something. When we were down doing field collections in Florida, some of the trees were so rotten that we could just take apart with our very hands. Like something like this. We're looking at all the entrances and the openings. It's just like totally coming apart. It's been eaten to shreds by termites. And if we do enough digging, there they are. There's your carpenter ants. And there's a winged reproductive too. You see that one right there? That's well, it's either a male or a queen. I think queen. That just climbed behind here, but you see there's the brood being carried around. So the embryos. So yeah, like they wouldn't be the ones boring into these holes. As you can tell too, these holes are bigger than our ants. So those are the hole sizes for some of those bigger termites. So we've got, yeah, a whole little ant nest underneath here. There's another reproductive. It's at roughly that season, y'all, where we'd start getting them wing reproductives. This is another reason why like a vial would have been fun to get. We could attempt to catch one of these and see if she's mated. The way we'd be able to tell is if she starts laying embryos. So as you can see, not our termites, but the ants have already moved in. And they're moving back into those holes in the nest, deeper in the nesting site. The white things they're carrying are the embryos. It looks like those look like larvae to me. Here we go. There's a view. You see how that ant is just carrying one thing? That one thing is a larva. So yeah, they've hatched and they're being fed now in development. Then it'll become a pupa and then it'll hatch into an adult. So 2 hatchings total. One from the egg and then one from the pupal case. Yeah, and then see, this is like the main entrance, nest entrance. That's where they're going. There might be another entrance down here too, but presumably underneath this rotting wood, there's another like tunnel network. Also, wouldn't surprise me if there were multiple colonies here in this tree. It's just so rotted away. There's no reason why a couple of colonies haven't made their home here. But there y'all go. 2 winged reproductives, if not 3 that we've come across. The embryos too. Look at that. See, pretty active. You just have to know where to look. And then it's not surprising that you don't have termites once you know what you're looking for. And then we've got some on the basin too of the ground. So there's some reproductive activity down here too. I saw a winged one and then they're carrying other brood around down here. But as you can see, that may very well be from just a different colony. It's just such a big tree, y'all. What's neat on these, y'all, there's fruit flies here. And you might be like, Balint, why on earth are there fruit flies on mushrooms? There are actually fruit fly species that specialize on fungi. And they can actually survive the toxins off some of these fungi. These fruit flies specialize on fungi that sometimes can be pathogenic to humans, meaning when you eat it, it actually shuts down protein synthesis in your cells. However, these fruit flies have a mutation that allows them to eat and get past it. So in the mutation in the protein is located right where this pathogen and toxin would bind on the protein. Is just different. And so the pathogen can't bind. And so the fruit flies survive. We actually did some research on those many, many years ago. 2 species, Drosophila fellini and Phalarotta. And then there's a close sister species called Drosophila palustris that is very closely related but is not resistant. I do want to see what these plaques are before we continue on our way out. Institute lands, 589 acre tract of woods and farmlands known for their natural beauty, historical significance, graceful woods and field key link between network of preserved open space between Stony Brook and the Delaware and Ravian Canal. Is a mixed forest with a northern sanctuary for migrating, nesting, and wintering birds. Huh, all right, there you go. 1997, advanced study along with public and private landowners, entities, over 1,000 individual donors created a permanent conservation easement on the Institute lands. Preservation of these historic and environmentally sensitive Institute lands was accomplished through this extraordinary community partnership. And then you've got all the sponsors here. And more sponsors on the plaques. Here, we'll even sit down next to this tree to make it look all like we're, one and beautiful with nature. Chat, thank you for hanging out with us today. I'm sorry Munchie was a little more fussy today than usual, but I hope y'all enjoyed it either way. We'll come back to here at some point because I want us to do a couple of interviews with some of the labs at Princeton. So we'll do that. And then we'll also check out some of the museums next time. Yeah, guys, thank you all for hanging out. But I'll see you on Discord and all the places there.